Thank you for the introduction. So I'm going to talk about a project that was actually partially conceived in the last ISMB in Long Beach, California, and so I'm very happy to be here presenting it today. And this is a method that I developed along with Uri Shalit and Gal Chechik from my lab. Using this method, we're able to learn functional representations for genes, and we can do this based on many types of biological data, and in this case, we're using a very special set of neural in situ hybridization images. So we're talking here about gene expression in the brain, and as we all know, genes control the development, structure, and function of the brain. Now, usually when people look at genomic expression patterns, they use methods that average expression over regions. So these are methods such as microarrays or RNA-seq. Now, when doing this, we mix up information from many different cell types, and also we use information that comes from small localized patterns of expression. So the Allen Institute for Brain Science tried to deal with this issue by collecting a genomic collection of neural in situ hybridization images for mouse brains. And in situ hybridization, as you all know, is a method that is able to capture gene expression in a tissue in a very high resolution. And let's take a look at the data. So this is a mouse brain. And you can see here gene expression for one gene. In this case, it's the gene ADAR. So black dots here in the image are our expression of this gene. And so each image shows expression for one gene in a cellular resolution. And here's another example. This is the gene GRIA2. So the images at first look, they seem pretty similar, but if you take a second look, you can see that they are sometimes tilted, and some regions look different in different brains. Each gene is sampled on a different individual mouse. So you can see the cortex here looks very different from the cortex here. And some images have little artifacts, such as this bubble here. So these are differences in global patterns, but the images have many small patterns of expression. So some patterns are more structured and easily interpreted, such as the layers here of the cerebellum or this ventricular zone here, but most patterns in the brain look like this, and these patterns are much more difficult to interpret. And also you can see that even things like tiny cell shapes look very different in different regions of the brain. So and these are just two images that we see. The collection of images is genomic, so you can imagine that there's a huge amount of spatial information in this collection. Now, usually when people look at this collection, they look at one or a few images for their own genes of interest. And looking at this data set as a whole genomic collection is a huge challenge, and it requires some computer vision. And so we wanted to do just that, and we asked two specific questions. The first one is, how can we identify similarities between these complex images? So we want to use a method that captures these small local patterns of expression that we discussed. Another question that we asked is, what makes these images similar to each other? Once we find the similarities, can we label them? So in a way, we want to make this problem into a more kind of a supervised problem than an unsupervised one. So our goal here is to explain the similarities in meaningful biological terms. So before we delve into our work pipeline, I'll just quickly mention that we didn't use the images as you saw them before. We used images that were masked by an algorithm to show only expressing pixels. And these images are also available for download at the Allen website. So using these images eliminates quite a lot of noise in our data, because we're only interested in the expression itself. So this is our pipeline, and it consists of two main phases, a visual phase and a semantic phase. So at the end of our visual phase, we will have a visual representation of the image, and in the end of the semantic phase, we will have a semantic representation of it. So the visual phase starts with extracting SIFT descriptors in 4 scales. Now, SIFT is short for Scale Invariant Feature Transform, and it's a very commonly used method to extract local visual features from images. Usually it's natural images. And there are many ways to extract visual features from images. SIFT has some advantages. It's invariant to rotation and illumination. So here's what it looks like. So we overlay SIFT descriptors on the image in an overlapping grid. And then we're left with a collection of descriptors that capture small-scaled spatial patterns based on local gradients of intensity in the image. And we repeat this step with 3 smaller scales. So we're not using the scale invariance property of SIFT here because we want to capture information in different scales separately. So this is as opposed to natural images or video processing. Okay, so now each image is represented as a collection of SIFT descriptors, a huge collection. So we want a representation that is more compact but still captures these rich patterns. And so what we do here is define different types of descriptors, and we want to count for each image how many descriptors we have for each type. So what we do is first we create what's called a visual word dictionary. And here we find our types of visual words, of types of descriptors. So for each scale, we pull the descriptors from all the images, and then we cluster them using k-means with k equals 500. So this is our dictionary size. And then we define different types of descriptors, and these are different visual words. And then, after doing that, for each image, we can measure the distance of all the descriptors of the image to the cluster's centroids, and then assign them to the appropriate cluster. And then we can count how many descriptors from each type we have in each image, and represent each image as what's called a bag of words of visual features. So here's our image, and we have descriptors from different types. So for example, the red type here, we count how many we have of this, and then we create this histogram that describes the number of visual words that we have in the image. So this is called a bag of words representation. So at the end of the visual phase, we have a visual representation where each image is represented as a 2004 long feature vector. So we have our dictionary size, which is 500 times the 4 scales, and then we added 4 extra features that count as 0 descriptors. So these are the descriptors that showed no expression. So you can say it sort of measures the sparsity of expression in an image. Okay, so here are some examples of real visual words mapped back on an actual part of an image, just so you know how it looks like. So each color here represents a visual word. So you can see that the green descriptor here captures these thick diagonal lines, and it does that for in different parts of the image. Okay, so this is our data. Now, at the end of the visual phase, we have our visual representation, but we want to add interpretability. So we want to get to a situation where we have a semantic representation of the image. So this is our semantic phase. And what we do here is apply semantic term classifiers to create a semantic representation. So specifically what we do is we learn logistic regression classifiers for 2081 gene ontology categories. So we learn each classifier separately. So we have a classifier for axonogenesis, one for RNA metabolism, one for translation, and so on. We use our SIFT bag of words or visual representations as input for the classifiers. And our positive set would be genes that are known to be annotated to these categories. So we used biological process categories with 15 to 500 annotated genes, since we don't want the categories to be too general on one hand, but then on the other hand, we want to have enough positives for our train test and validation sets. So the output of a logistic regression classifier is a probability for an image or a gene that is imaged to belong to each Go category. So now we can represent each image as a collection of probabilities for the gene in the image to belong to all Go categories, and this is what it looks like. This is our final representation, and this is, for example, an input to our algorithm, an image for GRIA2. This is a raw image, not a masked one. And then the output is a collection of probabilities for GRIA2 to belong to the category axonogenesis, translation, and so on. Okay, so now that we have our functional representations, we can first validate them and check for their stability. We can use them to find explainable similarities. This was our goal. We can also use them to infer new gene functional annotations, and we can also look at discriminative spatial patterns. So at first, we started with validating the results of our classifiers. So we wanted to see if we can actually predict functional annotations from these images. So it really wasn't clear to us if we'll be able to do that. Certainly, you can't see anything by eye with these images. They're so complex. And we used a bag of words representation, which discards spatial information. So we weren't sure that this would work. So we looked at area under the raw curve scores for our 2081 categories. And we were very happy to see that the vast majority of categories classified above random, above 0.5. And then lots of categories classified with really very high AUC scores. And then we asked which functional annotations can be predicted better from this data set. So since we're talking about brains, we hypothesized that categories that are related to neural functions, such as GABA metabolism or neuron recognition, would classify better. And indeed, when you look at neural-related categories, which is the red here, they classify with much higher AUC scores. So this makes sense. Okay, so now that we have our functional representations and we validated them, we believe them, we can very easily define similarity scores and check for similarities between the images. So we defined two similarity scores. The first one is flat similarity, and this is simply the dot product of two functional representations. So this measure is pretty intuitive because we're talking about probabilities, so it's basically the sum of the joint probabilities for the two images to belong to the same Go category. So while this measure is very simple to understand, it doesn't take into account the fact that the gene ontology is structured as a hierarchical tree. So we wanted to also insert this into our analysis. And we used another similarity score, which we called here Go similarity. And this was developed by Schlicker and colleagues in 2006. And this similarity score takes into account the hierarchical structure of the Go tree. Basically, it assigns more weight to categories that are smaller and more specific and thus more informative. So now that we have our two similarity scores, we can use them to look for explainable similarities. So the explanations for similarity would simply be, after calculating a similarity for two images, it would be the Go categories that contributed the most to the similarity. So now we have semantic explanations as well. Okay, so I showed before that brains for different individuals are different. And we wanted to try and assess the stability of our functional representations. So we wanted to check basically the amount of biological noise and technical noise in the data. So luckily we had around 740 genes that were imaged more than once. So this allowed us to check just that. And we looked at similarity scores for pairs of images that come from the same gene and then random pairs of images. And of course, same gene images got much higher scores than just a random pair. So that was a good sanity check. But the real question is, how does our method compare with a previous method that was designed to find similarities between these images? So this method is called a neuroblast, and it was developed by the Allen people. It's available on their website, and it's based on a mapping of the images to a reference atlas, and then a nonlinear spatial correlation between the images. So it's a very different method, and we used both methods to try and identify cell type markers for neural cell types, neurons, astrocytes, and oligodendrocytes. And here you can see a precision at top K scores. These are the top K similarity scores for pairs of images. And as you can see, FuncISH Go similarity performs much better than other methods. So it performs better than Neuroblast, the spatial correlation, also much better than the flat similarity score. So there is meaning to adding the hierarchical structure of Go to our score. And we also tested against our visual representation, the SIFT bag of word representation, which is the red line here. So we see that we predict cell type markers with very high precision. And we also had some success with predicting inclusion in the same KEGG pathway and protein-protein interactions, physical ones that we got from INTACT. But the real success we had was with cell type markers. And as an example of this, we looked at 3 genes that are markers for GABAergic interneurons. So calbindin, parvalbumin, and calretinin. These genes have been imaged more than once, each around 10 times. So they've been sampled a lot. And almost all of their pairs of images got very, very high scores when using FuncISH, but not at all when using a Neuroblast. So the similarity was not picked up at all by spatial correlation. And the simple explanation for this is because these genes are, they are expressed in the same cell types, but in completely different neural regions. So this is exactly the type of case that our method would capture very well, and spatial correlation completely misses. So another advantage of our method is that we're able to explain the similarity, to put labels on it. So when you look at the top explanation for the similarities of these genes, we see processes that are related to synaptic plasticity and learning, and this also makes a lot of sense for GABAergic interneurons. So it's easy to see how we can use the explanations for similarity for functional inference of new gene functions, and we have some nice examples of this in our paper. But I think that the really cool thing about this work is the fact that we're able to map the descriptors back onto the images, and then we can really see the small-scale discriminative patterns. And I think it's a new way to look at these images that nobody tried before. And here you can see the 3 most discriminative visual words for each scale, descriptor size, basically. So you can see that there's completely different neural information captured by all the different sizes. So the larger descriptors capture complex patterns of mixtures of cells in a tissue, and smaller descriptors capture thin cell lines. And the smallest one actually capture single cells and their shapes. So when looking at these descriptors, it seems that the larger ones contain a lot more information than the smaller ones. But then when we looked at the mean absolute weight for the descriptor sizes, we saw that actually it was the smaller ones that carried the most information for classification. So these descriptors that don't look like anything special to me carry a lot of functional information, apparently. Okay, so to conclude, FuncISH provides an interpretable measure of similarity between highly complex images, and we're able to successfully infer thousands of new gene annotations. Our method performs better than a spatial correlation-based method, and also we don't need any type of a reference atlas, so this is an advantage many times. Okay, so this is a joint work with Uri Shalit and Gal Chechik, and I'm thankful for the International Society for Computational Biology for providing me with a travel grant and enabling me to be here today. And thank you for listening. I'm happy to take any questions.
Questions? Did you
So you're asking why did we pick these? If we filtered out, yes, we use categories that have between 15 to 500 annotated genes. So yes, we didn't take categories that were too big and not too small, just because we needed some genes for our positives for different sets.
Do you think that the number of genes in categories may cause a bias in the results?
So we checked actually if there's any type of a correlation between category size and AUC scores, and we saw absolutely no effect. But we did check that.
How big was your sample size, and did they all come from the same institute, from the same population of mice? Or?
Yes, it's a very good question. So our sample size was, we had more than 20,000 images, but then more than 20,000, but then around 20% of the genes are completely not expressed in the brain. So these images were completely black. We discarded them, so we were left with around 16,000 images corresponding to around 15,500 genes. So this is our sample size. Now, all these images come from the same place, which is the Allen Institute for Brain Research, and the mice are genetically identical as much as they can be, and they're all sampled at day 56, and...
But per gene you had one image?
Yeah, no, but each image was one gene, and some genes had more than one image. They've been imaged more than once.
So you, in the final version that you do these SIF descriptors, and you completely throw away any information from where that actually comes from.
Yes.
And I quite like this myself. But my question is whether at some point you tried taking into account information.
Yeah, we did. It's a good question, and I have a slide for that question. I was prepared for that one. Yes, okay, but we didn't discuss it before. This is a coincidence. Okay, so we did try a spatial pyramid approach. So we didn't have for each pixel an annotation for a region, so this is the best we could do. So spatial pyramid, you just split the image into several parts, and then you calculate the dictionary separately for each part. And then what happens in this case is that your feature vector gets inflated, right? Because you don't have one dictionary or four dictionaries. You have 1 plus 4 plus 16, and it inflates. So we did look at our results, and they were inconclusive. So we figured that the increase in dimensionality hurts more than the gain we obtained by using some spatial information here. But great question.
So, quick question, how did you choose the number of clusters within the k-means?
Yes, I also have a slide for that question. Yes. Okay. So we did try several dictionary sizes. Okay, so we tried with the k equals 100 and also 1,000, 200, 500. So these are the mean AUC scores that we got with using the different dictionary sizes. You can see they're very similar. We had a slight increase with k equals 500, so we used that one, and also we checked and we saw that there's very high correlation between the AUC scores with k equals 100 and k equals 1,000. So I guess it doesn't matter that much.
For the SIF features, you said like 500 times 40, if I remember correctly?
For the scales?
Yeah. That what meant?
Yeah, well, we just used 4 scales. We tried using a smaller one and a bigger one. So each scale adds more information and computation time, right? You can see here that the most information is in this scale here. So since we got this sort of increase and then decrease, we figured maybe we shouldn't take smaller or larger scales.
Any more questions? Perhaps one short one. Personally, Who's the one?
I can ask a short question. What was your raw data format that you received the mice images in?
What's that again?
What was the data format that you received the mice images in?
The data format?
Was it just JPEGs or was it
yeah, and then
more
you just
sophisticated?
to transform it into